this course.
Good afternoon, everyone. Welcome to Native Voices in STEM, which is hosted by the Department of Chemical and Environmental Engineering for the spring of 2022. We have our third speaker here today, Dr. Otakuye Conroy-Ben, and this is the third speaker for a series of 4 for this semester. And the Native Voices in STEM is a program series that was developed under the Indigenous Food, Energy, Water Security, and Sovereignty Program, which is funded by the National Science Foundation. We currently have 38 graduate students who are learning about transdisciplinary research and collaboration across the food, energy, water nexus while interacting and doing work with Indigenous communities. So we have a variety of components to that training, including immersion, training at tribal colleges, seminars, and other workshops, and the Native Voices in STEM. Native Voices in STEM is now institutionalized at the University of Arizona and is operated and run by the Office of Societal Impacts and hosted by different departments every semester. So I'm really pleased to have Dr. Otakuye Conroy-Ben here with us again. She was previously featured in our Native Voices in STEM, and we're really gracious to have her come back again and be part of the series in particular because she graduated from this department, Chemical and Environmental Engineering. So it's wonderful to have her speak about her work that she does. And so at this time, I'll give it over to Dr. Sarah Abney, who is one of our Indigenous youth trainees and recently defended her PhD. And I'll have her introduce Dr. Otakuye Conroy-Ben further. Thank you.
Thank you, Dr. Chief. Just to give a little introduction to our esteemed speaker today, Dr. Otakuye Conroy-Ben is an assistant professor in the School of Sustainable Engineering and the Built Environment at Arizona State University. Dr. Conroy-Ben has received a BS in chemistry from the University of Notre Dame, a master's in analytical chemistry from the University of Arizona, and a PhD in environmental engineering from the University of Arizona. Her research focuses on the biological effects of polluted water, and her interests include environmental endocrine disruption, metal and antibiotic resistant in bacteria, and wastewater epidemiology. So with that, I'll turn it over to Dr. Otakuye Conroy-Ben. And thank you again.
Okay, thank you for the introduction, Dr. Abney. Let me start my slideshow. And the title of my talk is Wastewater-Based Epidemiology in Tribal Communities. I want to kind of talk about wastewater as a whole because that's where I started out at the University of Arizona, and all of the research experiences and projects have really led up to this type of work. And so, as stated, I got my master's and PhD from U of A under the direction of Dr. Bob Arnold. This was in the early 2000s when I started looking at wastewater reuse and the biological effects of wastewater impacted waters. But this slide pretty much summarizes, my wastewater career to date. If you look at the center, this should look familiar to those of you in the audience down in Tucson. This is the Sweetwater Recharge Facility at the former Roger Road Wastewater Treatment Plant. This is what I did my graduate work on. We're looking at endocrine disruptors, estrogens and androgens present in wastewater effluent, and looking at how different processes such as infiltration into basins, wetlands treatment, as well as just transport along the Santa Cruz River changes the signal of estrogenic and androgenic endocrine disruptors. After graduate school, I worked at LA County Sanitation Districts, so in the lower left. I worked specifically at the Joint Water Pollution and Control Plant, figuring out ways to mitigate odor coming from different wastewater operations, such as sewer transport and biosolids handling. Also in there, I had a chance to work with the National Congress of American Indians as an intern, where I evaluated the environmental impact statement of the Snowball Resort that was reusing wastewater effluent as part of their snow production. Currently, I'm doing a number of different types of research, looking at the fate and transport of organic pollutants coming into facilities, leaving facilities through the liquid streams, as well as in the biosolids. A few years ago, I had the opportunity to travel to New Zealand and work with a Maori community where they were experiencing a significant amount of pollution coming from wastewater effluent that was discharging into one of their cultural and significant lakes. I know that there are students in the audience at different levels. This is kind of where my career has taken me. Starting as an undergrad, I participated in research, which really piqued my interest. So I ended up applying to graduate school, ended up at the U of A, and had a great time there with the different programs, and I learned quite a bit, and had some truly outstanding mentors there. When I graduated, I worked at LA County. I was there for 1 year, and then I decided that I wanted to get back into academia. And so I returned to the U of A and was a postdoc fellow in 2 programs, biochemistry as well as environmental science. I'll talk more about that work shortly. Also, as a postdoc, I was an adjunct faculty member at Pima Community College. I then moved to the University of Arizona, and then after that to Arizona State University, where I have my position. And so I wanted to kind of summarize my research, because what I did, over 20 years ago is really come full circle, and I'm doing this today. I was part of a summer research program for 2 summers, and one of the labs ended up hiring me after I graduated with my bachelor's. And we were looking at the genetic bases for certain responses to alcohol when an organism ingests alcohol. And in this work, I was able to learn about sequencing, DNA sequencing, and we were sequencing some mouse genes. This was back in the late 1990s, where the technology was novel at the time, but compared to today's sequencing improvements, this is really quite ancient technology. What came out of this work was, an appreciation for research and learning new methods, as well as, it gave me a boost in publications, which really helped me get into some pretty strong graduate programs. And I ended up at the U of A. And this here, I would say, summarizes my graduate work at the U of A with Dr. Bob Arnold. We were looking at endocrine disrupting chemicals. At the time, in the early 2000s, these were pretty novel pollutants. It was known that fish living in wastewater impacted waters were experiencing some interesting side effects, such as intersex, male fish acquiring female characteristics, and so forth. And it was due to the presence of mainly organic pollutants present in wastewater. These pollutants were not getting removed during the wastewater treatment process. They weren't getting removed completely, and they were still present at concentrations that were biologically relevant. And so, we were interested in looking at removal during wastewater treatment and what happens to the endocrine disrupting chemicals once this wastewater is discharged into the environment. We used a couple biological assays shown on the left, which measures these chemicals. We measured the different chemicals and their responses in this assay. We looked at the Santa Cruz River, tracking the estrogenic signal along a length of the Santa Cruz River, as well as in wells lying along the river. And we showed a correlation between wastewater content as well as estrogenic chemicals present in wastewater. So I found this work to be, very exciting and novel. And with that, I knew I wanted to stay in research. And I ended up as a postdoc in the labs of Dr. Megan McAvoy, who was in, at the time, chemistry, biochemistry. She's now at UCLA. She is a protein biochemist who looks at metal homeostasis in gram-negative bacteria. Was also in the lab of Dr. Chris Rensing, who was in environmental science, and he studies this as well, metal homeostasis in different types of bacteria. So as a postdoc, I wanted to get new experiences. So I was excited to join this project where we were looking at the interactions of a metallochaperone called CUS-F. We're looking at the interactions of this protein with a metal exporting protein, CUS-CFBA. When E. coli experiences high toxic levels of copper-1 or silver-1, they will transcribe this protein, enabling for export of copper from the cell. So basically, it's a response mechanism to intracellular toxicity. And this is a way that bacteria can survive in the presence of high metal concentrations. We found that there are these proteins present in E. coli, in salmonella. Salmonella can export gold. And C. metallodurans also has a number of proteins that can export cobalt, nickel, and cobalt, zinc, and cadmium. This is called metal resistance. And it's actually very similar to multi-drug resistance in bacteria. And so why is this important? Throughout my career, I wanted to see how this can be applied in engineered systems. And what is the relationship between antibiotic resistance, gram-negative bacteria? What are the roles of these proteins in environmental engineering? So early on, I recognized this relationship between metal resistance and multi-drug resistance. And if you look at the proteins, these proteins span the inner and outer membrane of bacteria for metal resistance as well as multi-drug resistance. And if an antibiotic is present in a bacterium, it will be exported from the cell. This is an energy-driven process, allowing for survival of bacteria in harsh environments. So E. coli is my model mechanism. In the previous slide, it does have some resistance to copper and silver. It also has quite a bit of resistance to antibiotics. They have, E. coli has 7 of these different multi-drug resistant pumps present, which allow it to survive in harsh environments. They can export antibiotics, biocides, metals, hormones, detergents, and so forth. Even more noxious would be Pseudomonas aeruginosa, which has about 20 of these different protein complexes, really allowing for it to be resistant to most antibiotics out there. So, how does this relate to environmental engineering? Well, environmental engineering, we know that the biological reactor in the treatment plant, which is required by EPA, must remove at least 80% of organic matter. And I got to thinking, well, if these efflux pumps are present in the bacteria that are basically required during wastewater treatment, these proteins may be limiting that biodegradation process. So I received some NSF funding to look at that. We characterized all of the different efflux pumps present in wastewater treatment using metagenomic sequencing, as well as their removal, the removal of these antibiotic resistance genes through wastewater treatment. And all of this work, really does support our hypothesis that the presence of these efflux pumps does limit biodegradation. So I partnered with a chemical engineering professor here at ESU, who is a metabolic engineer. And we grew up E. coli in C13 labeled media. So basically every carbon in this E. coli was C13 labeled. And we exposed the bacteria to the different emerging contaminants, such as ethinyl estradiol, BPS, BPA, to see if the presence of these pumps prevents degradation. And so we're looking for changes. We're looking at the amino acid fingerprint. We're looking at the carbon 13 and carbon 12 signal of these proteins. And basically, what we found is that the presence of these efflux pumps, these antibiotic resistance pumps, forces chemicals out of the cell. And the absence of these pumps forces these chemicals, these emerging contaminants, into metabolic pathways. And this is really important when it comes to that biological reactor, where we're not seeing complete removal of emerging contaminants during wastewater treatment. And one of the possible mechanisms for this is the presence of multi-drug resistant and so I'm continuing to do that related work on emerging contaminants, biodegradation during wastewater treatment, and then the fate of antibiotic resistance genes, as well as chemicals in wastewater treatment. I did speak to this audience a couple of years ago as part of the Native Voices in STEM before the pandemic. So some of you may have seen this work before, but Dr. Carletta Chief led a special issue in the Journal of Contemporary Research Education, I want to say. And she invited authors to submit some work. So I'm showing you now some tribal work. Looking at Safe Drinking Water Act violations. Basically, what my group uncovered was that there are more Safe Drinking Water Act violations in tribal public water systems, where we have violations of MCLs, but more likely it is due to monitoring, reporting, and public notices. Also in another special issue led by Dr. Carletti, I looked at the unregulated contaminant monitoring rule and the prevalence of these chemicals in tribal public water systems. And if you're not familiar with UCMR, EPA has to monitor 30 unregulated contaminants every 5 years and measure these chemicals nationwide. So every 5 years, they come out with a new list. They measure concentrations as well as how many people are impacted by these chemicals. So these aren't regulated under Safe Drinking Water Act, but they could possibly be regulated at a later date. And looking at the data, we saw that under UCMR 1 and UCMR 2, that these chemicals were not detected in tribal public water systems. But starting with UCMR 3 and UCMR 4, a number of different organics and inorganics were quantified in tribal drinking water systems. Currently, EPA is monitoring under UCMR 5. Under UCMR 5, they're looking at 29 PFAS chemicals as well as lithium. So it'll be interesting to see how if PFAS is found in tribal communities. But related to that work, I'm looking at some molecular docking. So we took all of the UCMR chemicals and measured their binding affinity to the different endocrine disrupting proteins in major pathways, such as the estrogens, androgen, progesterone, mineralocorticoid, glucocorticoid, PPAR gamma, thyroid, to see if these chemicals are endocrine active. And we also looked at the water prevalence data to see how widespread these chemicals are. And this is just a summary of these results. The molecular docking did take some time. It took about 1,600 computation hours. But everything is summarized here. And we can total the binding affinity and relate it to a net toxic potential. So in the plot on the right-hand side, we have a toxic potential for endocrine disruption ranging from 0 to 1, and the number of public water systems that these chemicals were found in. And you'll see a range. We have some highly toxic chemicals that were found only in a handful of United States public water systems. Then we also have a number of weakly toxic organics that were found widespread across the US. So what does this mean? Well, these chemicals are being evaluated by EPA, not only for endocrine disrupting potential, but for other biochemical pathways. And this is just one aspect that we use to evaluate the potential toxicity. But I really want to focus on how all of these research experiences led me to this my large project, which is wastewater analysis of SARS-CoV-2 in tribal communities. This is NIH funded. We applied when at the start of the pandemic, federal agencies really wanted to get a handle on the pandemic. And one of the RFAs called for wastewater-based epidemiology as a tool to monitor community level infections. So as a wastewater person, as someone who had previously done this type of work related to substance abuse, this was an opportunity to bring some novel monitoring and methodology to underserved communities. And so I partnered with a number of faculty here at ASU, as well as with the Intertribal Council of Arizona, to look at wastewater-based epidemiology in tribal communities. This specific project, we propose to recruit 5 to 10 tribes nationwide for monitoring, as well as to look at coronavirus levels in wastewater, to look at protein present in wastewater from SARS-CoV-2, to assess risk, as well as to optimize the viral monitoring strategy. This slide basically summarizes all of the work. I'll get into the detail. Our minority populations have been adversely impacted during the pandemic, as you are well aware of. And in particular, American Indian, Alaska Native have been the highest case rate as well as death rate during many of these phases. And as you know, someone from a tribal community, I see how it's impacting, you know, loss of our elders, loss of that traditional knowledge. It's really impacted tribal communities adversely. And when it comes to wastewater-based epidemiology in tribal communities, we're dealing with different infrastructure. I've been to a number of different communities, sampling, speaking with health departments, utilities, and it's a concept that is, you know, well received in with some tribes, and some other tribes don't really see it as being a benefit to contributing to their data stream. If you're not familiar with wastewater-based epidemiology, we're treating wastewater as a dilute sample of human waste. We're collecting both solid and liquid waste, and anything that's excreted in these waste products can be quantified with some sample prep, of course. Wastewater-based epidemiology, or WBE, can be used to measure substance abuse or substance use, contaminant exposure, as well as infectious disease. There are about 100 tribes that have centralized sewer treatment in the US, and samples can be collected from different points. But when it comes to tribal communities, most of the tribes we work with have very basic infrastructure. It's rural, smaller flow rates. And so we're typically dealing with lagoons, shown in the middle. Lagoons are, you know, a large body of water, and the coronavirus signal is pretty dilute. And so we actually have to collect from manholes in some cases. The advantages for WBE: we can rapidly assess community health. This is anonymized, it's non-invasive sampling. We're not requesting biospecimens, for example. Some tribes do have moratoria on that. Some disadvantages: sewer contains human pathogens, and it's encouraged to get additional immunizations. We have to use PPE to limit exposure to bacteria and viruses. Population estimates are not well cataloged, as well as the sewer flow rates. In wastewater-based epidemiology, we take our concentration level and we would normalize it to the flow rate as well as the population. How we collect our sample: we have an ISCO portable auto sampler that is either lowered into a manhole, or if the tribe already has this infrastructure shown here on the left, we would collect samples every hour for 24 hours and analyze each sample, also make a composite of that sample, and quantify it for coronavirus or whatever other measurement we would like. last week, I was up in the Northern Great Plains sampling for one of the tribes, and it was 5 degrees out. It was incredibly cold. It was not fun trying to pour off sewer samples in that frigid weather. So in aim 1 of this NIH grant, we were tasked with looking at tribal wastewater infrastructure. Of the 574 federally recognized tribes across the US, 326 have a land base, meaning they have a reservation, or some type of documented property where, in theory, there could be people living there with, which would require sewer infrastructure. Of those with a land base, 105 tribes have NPDES permits according to EPA ECHO database, which means that there are 221 other land-based tribes with no wastewater permits to discharge. That doesn't mean they don't have wastewater. These tribes could not be discharging. They can use their wastewater effluent for reuse, or else the facility itself is a total retention lagoon, meaning it just won't discharge. And we use this as a starting point to start to reach out to tribes. Do they have the right infrastructure to do this type of work? And who are these tribes? What is their research process like? And who are their utility contacts? And so this map just shows you where these tribes are and which have the facilities to potentially collect wastewater from to do this type of work. You'll see on here there are some casino wastewater treatment facilities as well as school wastewater treatment facilities. There are also Indian Health Service facilities. The schools, some of these fall under the jurisdiction of Bureau of Indian Affairs, Bureau of Indian Education, which require additional levels of approval. And so we are not targeting IHS facilities as well as VIA schools. We know that there are a number of other tribes that do have wastewater facilities, but we're not showing up in the EPA federal database for NPDES permits. So my colleague here at ASU, Dr. Rebecca Munich, as well as staff engineers at ITCA, what they plan to do, and they're working on this now, is to look at using remote sensing to track wastewater lagoons within tribal boundaries. And in this way, they'll start to get a more complete list of these wastewater facilities present in tribal communities. Perhaps we can start to develop that list of the other 221 tribes that may have wastewater operations. So looking for tribes with wastewater facilities is the first step in doing this type of work. The next step is getting approval from our tribal partners to conduct this work. This requires going through either IRB processes or some other type of approval to do NIH-based research. And that was, it's required of ASU as well as of NIH to make sure that we have tribal approval to do any type of work. And this slide summarizes the different tribal approval processes to use wastewater-based epidemiology to look at community health. Since this project started a little over a year ago, we've inquired with about 20 tribes nationwide to see if they are interested in doing this type of work. We've submitted 12 tribal applications, and if you've been involved in any type of tribal IRB or presenting to tribal council. This can be, a large task. You're basically presenting a proposal to the tribe. Given that we're in Phoenix, 11 of these tribes are in the Arizona area served by Intertribal Council of Arizona. When we submit an application, 4 of these applications went through an IRB process. So there's an IRB board. 5 of our applications went through a different process where we present to the health committee of the tribe, who then recommends it or does not approve. And if it's recommended, we then present to tribal council, who would then either approve or not approve, and then write it into the tribal resolution. In some of these applications, there are other processes, such as a research permit, or just inquiring directly with tribal council. We have some interest from tribes where they actually don't have a research policy in place yet. That's where you would see no process. And in this case, we did not move forward with working with those specific tribes, simply because it's important that the tribe, protect the data that is generated from their boundaries. And it's also important just to know as academic researchers, what policies are allowed and what are not allowed. When we submit a tribal application, we've received a decision within a few days or a week, and in some cases up to a year later. A couple of the tribes, I'm still waiting on a decision. On average, it takes about 3 months from the application to get some type of approval. Tribes have responded that, yes, this is innovative. We like it. It's non-invasive. You're not asking for tribal members specific biospecimens. And so, in that way, some tribes have been very receptive. Where we've received answers of no, as stated earlier, some have thought that this type of work will not contribute to the COVID data collection stream. A couple of the tribes have had some complicated water rights, which would require approvals from multiple entities. And some tribes have just not been interested. And so the tribal approval process, is incredibly important. It's time consuming, requires patience. You have to develop those relationships. I'll show some data later, but we've actually started wastewater-based epidemiology with tribal communities back in 2016. And so we have already had these relationships ongoing. It was just a matter of looking for a new analyte once the pandemic started. I've had a lot of learning experiences in working with tribes in this approach for health monitoring over the years. Realized that tribal approvals may involve IRBs, resolutions, MOUs, MOAs, MTAs, the application and the proposal. We have an ASU Tribal Research Review Office to ensure that academic researchers are following all procedures, tribal as well as institutional. And so they are present at all stages and with our meetings with tribal communities. We coordinate with the tribal utility as well as with the health department. These are some interesting conversations. We're bringing 2 different programs, departments together who may not be in regular communication. Tribal utilities are severely understaffed, and we have funding for tribes to do this type of work. However, due to staffing, myself, my students, we are out there sampling. Sewer access is limited. If you're in a remote community, staff is, don't have enough staff, we may not have access to collect samples. And so it's important to keep that in mind as well. Sample location, collection, and delivery are challenges. For sampling in the Northern Great Plains, you have to fly there, drive several hours, drive it to a FedEx several hours, and then FedEx that back here. So it's, it can be up to 4 days before the sample reaches ASU. ASU, myself, my research team, we recognize tribal data sovereignty. And so any data generated is returned to the tribe. The tribe owns the data. And so as researchers, you must understand reporting and dissemination policies outlined in the tribe's research program, and also pay attention to what is required for federal reporting. So, I've had a number of challenges. The Office of the President at ASU, they have a number of faculty and staff who assist researchers when working with tribal communities, but also really key in this NIH program of wastewater-based epidemiology is Intertribal Council of Arizona, who have that, they've already developed the relationship. They know the tribal policies for all the Arizona tribes. They also have an environmental program where they're in regular contact with tribal wastewater operators. And that's under the direction of Travis Lane and William Brennan. So looking at coronavirus. I'll try to speed it up the last few slides. We are analyzing both for SARS-CoV-2 gene quantification using qPCR. Samples are returned back to ASU and processed within 24 hours so that we don't lose that signal. Okay, and we also propose to look at the proteins of coronavirus, and that's because we started to see coronavirus signal, decaying after the long transport time, as well as due to high temperature during transport. Another aim under this project is to look at the signal during sewer transport. So we're looking at the Tempe sewer shed, measuring coronavirus signal, looking at different factors that would impact that. Also, Dr. Carrie Hamilton, who is a microbial risk assessment faculty member, she's developing a risk model for occupational exposure due to wastewater handling. In tribal wastewater facilities, many of these lagoons are not disinfected, and so it's possible to be exposed to other pathogens other than coronavirus, to be put at risk due to exposure. Outreach, dissemination impacts. We have 2 data dashboards under development. We'll be conducting a tribal wastewater-based epidemiology training program in May. And our tribal partners have expressed interest in how do we handle the data, how do we sample, and so there'll be different components of this available. And if you are interested, please reach out to me. And we'll also be working with the Inspire Summer High School program to show them this type of work. We've had impacts in tribal communities. The town of Guadalupe here, which is encompassed by the city of Tempe and is home to Pasco Yaqui. They use wastewater-based epidemiology to cut its COVID-19 cases early on in the pandemic. This is a dashboard created by Dr. Rolf Halden, another co-PI on the project, where you can get data, weekly data on coronavirus. And this week or next, we have a wastewater-based epidemiology paper coming out looking at substance abuse monitoring. And this just summarizes some of the data in that paper. Other wastewater-based epidemiology projects, I'm partnering with ITCA. We're looking at relevant biomarkers present in tribal communities. So we're going to develop a tribal wastewater-based epidemiology framework. We'll evaluate tribal perspectives and explore wastewater-based epidemiology in tribal communities. Under another grant, I'll also be sequencing the variants present in tribal wastewater using PacBio. I'll be using the instrumentation down at U of A. And then we'll also be taking the sequences and doing some protein modeling. This is my team. Myself, Dr. Halden, and Carrie Hamilton are the PIs on the grant. Doctors Driver, Munich, and Dr. Joseph are doing the remote sensing, sampling, analyzing. Marcus is also working with tribal communities, and then we have our ITCA partnership. And I just want to say thank you. I also want to point out that I did not show any tribal data because our tribes have chosen to remain anonymous, and the release of the data is at their discretion. Thank you. These are all our partners as well as funding agencies. Or not? Yes.
Awesome. Thank you, Dr. Conroy-Ben, for the amazing presentation that you just gave us. We will segue into a Q&A just for students since we are a little over the time. So anyone here on Zoom in Harshbarger, students, it is a student-only Q&A, so please stay. Dr. Conroy-Ben is nice enough to give us more of her time today to field any questions that we have. But I would like to open it up right now just very quickly for the last 2 minutes if anyone non-student had any questions.
Hi, I'll start the Q&A with a question while people are thinking. Thank you so much for that presentation, Dr. Conroy-Ben. It's very interesting. I was interested in the higher pesticides that you found and the emerging contaminants in tribal communities and the lack of regulation of pesticides, and thinking about how agriculture is very important for many tribal communities, particularly those who do traditional agriculture. So I was wondering if you had any thoughts about how a tribe could think about pesticides in relation to, regulating it, but also the importance of regulating that in communities that have larger agricultural production.
So I guess it would be up to the tribal environmental or water director to get some measurements. If it's under these UCMRs or any other program like that, I know that EPA does have funding for that, or they can provide resources. And so, as far as, on the regulatory side, I think getting that baseline data, and then tribes do have a right if they have the state's tribe-to-state status tests to start to implement their own regulations. I don't know if that answered your question.
Yeah, thank you.
I'm a student, but I don't think I can ask a question, correct?
Good
You
afternoon.
Go ahead, Cara. Yeah, sure, you can ask a question.
Okay, it seems like your presentation was much more focused around the wastewater and everything, but is it much more centralized just to a certain community, or is it pretty much bridged along other communities across the reservation? Because there are multiple reservations across the United States, and wastewater and the contamination is a huge issue, does take its toll on a lot of the health of the residents within these areas.
Yeah, definitely. And in the community meetings for our tribal partners, that topic is always brought up. They are interested in what are the coronavirus levels, but they're also concerned about just wastewater discharge. And so we have a number of tribal partners and a number of communities where they're echoing the same concern.
The reason why I ask is because I live in a community where there's a large mine which recently closed, and there is speculation that the waste is impacting the water within the community. But due to nobody looking at XYZ, it's only XY, in and out. There's nothing looking at Z. And I question that as well, because when you taste something from the faucet, you can easily taste the amount of chlorine within it, or else some days it varies within taste, I guess you could say.
Yeah, so mining waste is different, right? It doesn't fall under, it can be considered wastewater, but what I'm looking at is the municipal wastewater. And for drinking water, I've also seen, violations of that in that report, but also from community newsletters where they say in these notices that your drinking water may not be safe for consumption at this time.
Awesome. Well, thank you all for the questions. Before we segue into just a student only Q&A, I did want to acknowledge our sponsors for Native Voices in STEM, which is co-sponsored by the NSF, NRT, and Digifuse at the University of Arizona, as well as the Sloan Indigenous Graduate Partnership, the Water Resources Research Center, and the Department of Chemical and Environmental Engineering. I'd also like to acknowledge that the University of Arizona is on the land and territories of Indigenous peoples. Today, Arizona is home of 22 federally recognized tribes, with Tucson being home to the Odom and the Yaqui nations. The University of Arizona is committed to diversity and inclusion and strives to build sustainable relationships with sovereign Native nations and Indigenous communities through education offerings, partnerships, and community service. Thank you again for attending Native Voices in STEM as a public open. We'll now segue again into our student-focused Q&A. For those who are interested in following Native Voices in STEM further, we do have our next Native Voices in STEM seminar on April 11th at 3 p.m. Mountain Standard Time, both in person and Zoom. Our speaker will be Dr. Tommy Rock, who is a postdoctoral fellow at Princeton in the Department of Geosciences and will be speaking on environmental justice. So thank you for everyone in attendance, and now we will segue into our student-focused Q&A. And thanks again, Dr. Conroy-Ben. Let us know if you'd like to take a break in between this Q&A session, or we can go straight into it, whichever you'd prefer.
Yeah, we can go straight into it. That's fine.
Awesome. Well, I'll leave it open if anyone would like to just speak up or message in the chat. One of us can also read your question as well.
Hi again, I'm speaking. I have multiple questions because I'm a mechanical engineer student at the Tohono O'odham Community College in Sills, Arizona. But still, there's multiple questions that I have, but I don't want to take up too much time asking my questions because of it's just, I don't know. But it seems like, is your project mostly focused just within one specific community right now?
No, so we have, right now we have 5 tribal partners, 5 tribes. I would say we're collecting from probably, grab samples too, not full samples. I would say we're probably around 15 different sewer sheds. So we're looking at least 2 communities being towns with each tribe.
I have a quick question. Can you guys hear me? Oh, awesome. Okay. Hi. I had a quick question. When you're talking about sampling lagoons, I think you mentioned how it's a more dilute signal. So I was wondering how it's different to sample lagoons versus like a centralized system.
Right. So we don't sample from lagoons because we haven't ever detected coronavirus in a lagoon.
Okay, so just centralized systems then.
So we sample from the sewer line right before the lagoon.
Oh, okay, got it. Thank you. Thank you. I enjoyed your talk. I had a quick question about oh, sorry. Okay, now you're oh, hi. Sorry. Regarding, I really like at the end kind of talking about tribal data and kind of having that autonomy to like have those results kind of within the tribal nations that you collected the samples and having that kind of sovereignty over that. I just had a question about kind of your experience introducing that into the Western academic lens, I guess. So kind of your experience with kind of as an Indigenous scholar, kind of in this Western research lens of prioritizing tribal autonomy when it comes to sharing those results. And yeah, hopefully that makes sense.
Yes. Yeah, so I guess when doing any type of research with tribes, all of that is pretty much laid out for you as a researcher, that the tribe owns the data, or if you wanted to share data, you have to get permission. If you want to publish, present, you have to get permission. And I think as an Indigenous person, you know, I've always understood that and respected it. I see that the federal agencies are understanding that more. NIH does have a policy that you have to report data except with tribes. And so I haven't had any issues when working with federal funding and with tribes.
Okay, I myself have a question now. So I've, done research and tested the effluents from wastewater treatment plants, and some that are heavily funded, like in Scottsdale and we're still finding, virus even in those effluents from point failures. So I was wondering, in what aspects does your lab look at, especially wastewater treatment, but then, what do you think as a researcher are ways that even treatment plants or we as researchers can really protect citizens from those point failures, or where we're emitting viruses into the environment, considering that you're looking in tribal nations, especially in comparison, to what we see as point failures in these like multi-million-dollar wastewater treatment plants. Have you seen a difference, and have you, used any public health interventions to either educate or maybe limit the concentration that's coming out in those effluents?
Yeah, so with respect to this particular project, we only have approval to measure coronavirus. So we're not looking at any other viruses in tribal communities. For one tribe where we looked at organic content, we did measure influent effluent. That was a chemical measurement. I take measurements here in Phoenix as well, but I'm only looking at bacteria. And I'm finding some breaches even in plants that do have fully disinfected effluent. And so I guess I can only comment on different aspects of it. I'll give you one example. There's a tribal community that went through some flooding, and it was a lagoon. So wastewater lagoon get untreated, undisinfected effluent, I should say wastewater, got out in the surrounding community. And the EPA ended up, I believe they got fined because there was a breach in that aspect. And so, how can we work with tribal communities to do that? Well, we need better treatment systems. Lagoons are very different than centralized wastewater facilities. We need to, as engineers, provide solutions for that. Perhaps build higher retaining walls or something. Making sure that information gets communicated out to the surrounding areas, as well as maybe just taking some measurements. What did get out there? If the tribe themselves, they can't do viral monitoring, they can look at some type of bacteria to show evidence of failure of the treatment process.
And I guess as a follow-up, has this project here, especially that's focused with COVID or SARS-CoV-2 within the wastewater, has this stimulated other potential future projects of other contaminants that are coming out of wastewater effluents for you and your lab, such as microplastics, pepper mild model virus, indicators, etc.?
Yeah. So yes, it has inspired. We've been monitoring for drugs in tribal communities for a number of years. And we want to look at other wastewater indicators. So with the recent NARCH funding, we're going to explore that. There was a tribe who was interested in also looking at the flu virus. Because there were, at one time there was talk of, flu, co-infection with coronavirus, or maybe they're off peaks during the year, and so forth. So it's just a matter of, prioritizing and then getting that approval again to monitor something new.
Awesome, exciting stuff. Thank you.
Hi. You may have already mentioned this, sorry if I missed it. Is the main purpose behind the measuring to kind of serve as like a first alert for COVID in the community?
Yeah, different research groups, including ours at ASU, have reported, the coronavirus shows up in wastewater before it shows up clinically in the population. Depending on which part of the pandemic we were in, that ranged from 2 days to 10 days. You would see, if you saw an increase in wastewater, you would see it 2 to 10 days later in the clinical population. We also saw that with sequencing. For Omicron, it showed up in wastewater before it showed up clinically in the US. It actually showed up before they reported the first US case.
And is there, is it expected that the measuring will continue for as far as the future goes? How long it'll continue?
Right, so NIH is funding this project for 1 more year. And they're really interested in seeing how wastewater-based epidemiology could be applied to future pandemics or to other health indicators. And so, this has been going on for 20 years, wastewater epidemiology, but it wasn't until the pandemic that it was really embraced by federal agencies for funding.
Okay, great. Well, with that, if we don't have any more questions, I'd like to give a big thank you and hopefully a round of applause with help of my cohort and other students for Dr. Otakuye Conroy-Ben. Thank you again for taking out the time from your day to speak with us and tell us more about your research and what research got you to where you are today. It's been really interesting. We actually do have a lot of overlapping research from undergrad to even my PhD. So thank you again, and thank you for everyone who tuned in today in person and over Zoom, especially to our co-sponsors in Digifuse, Sloan, the Chemie Department, and WRRC. Just another reminder and a little plug, Native Voices in STEM does have another presentation on April 11th, 3 p.m. Mountain Standard Time with Dr. Tommy Rock. So please tune in again. And again, thank you, Dr. Conroy-Ben, for today. And if everyone could just give a round of applause. Yeah, there we go. Thank you.